All right, so I wanted to make a video for you guys showing you how to open up this virtual lab. Again, these are from Michigan State University. They do an excellent job highlighting a lot of the stuff that we want to learn in this class. And so to get there, though, it's a little tricky. All right, so depending on whether you have Windows or Macintosh will determine a Mac. So that'll determine which of these you're going to click. Okay, but to open this virtual lab, this is what you need to do. All right, so let's go to Graham Stain. Actually, let's go to Streak Plate. I think that's the one I have you start with. Okay. You're going to click over here and download Flash Player, and it should pop up down here. Okay. The next thing you're going to do is once you get here, you're going to go and click on, oops, here. Once you get there, then you're going to run and click on this. You're going to click Download Streak Plate. Let me get this out of the way. And we're going to click Open. So it just opened that. Okay. It doesn't seem like it did, but it did. Now I'm going to go over here, go to Adobe Flash. I'm going to click File, and I'm going to click Open, and then I'm going to go Browse and find the one I just did. So whether it was a Graham Stain or whether it was a Streak Plate, I think is the one I wanted. I'm going to click Streak Plate, and I'm going to click Open, click OK, and it will run it for you. Okay, so just make sure that you do that when it comes time to have to should be able to open this all the way. There we go. Just make sure you do that when it comes time to actually start your virtual lab. And everyone will go that way. So whether you're doing the virtual lab for streaking or Graham staining, whatever it is, same process. Okay. Just make sure that when you go to browse, you can find the little file once it's downloaded. So we haven't talked about streak plating yet, but it's another technique for growing bacteria. We said that's where we were moving next, is learning how to grow bacteria. You guys know what a flame loop is. It's this guy right here. Okay. First thing you're going to want to do, we always flame this loop. We're assuming it's dirty. You see it get nice and red hot there. Take it out. And we're going to peel a colony off of this plate and put it on this plate so we can grow whatever was here onto here. Okay. And so the first thing is you want to look for an isolated colony. Okay. So like one of these guys, we can guarantee stemmed from one bacteria. This, really hard to tell what's going on here, if it's one bacteria, if it's multiple. So find an isolated colony, one little circle of, I don't know, a few thousand or a few million bacteria. So I'm going to grab this guy right here. I'm going to take that, open my new plate, and then you'll notice it's broken into quadrants here. You're going to take, you can see it's kind of a little yellowish color on the end of my flame loop. We are going to streak that. That's where the name streak plating comes from. This is how we grow bacteria oftentimes on Petri dishes, okay? Whether it's pulling from a colony on agar, or whether it's pulling from broth that you get. That's the stuff you guys did with your lab last week. Okay, so you're going to take this and you're going to drag this back and forth. Oop, I dragged into the wrong quadrant, so I must have went too far. So drag back and forth, but don't cross into the next one. Okay, then we want to thin out the amount of bacteria because we just pulled a few million. We don't want a few million growing necessarily. What I'm going to do is flame my loop and kill all of those bacteria. All good? Now, I'm going to take my sterile loop with nothing on it, and I'm going to drag from these that I've already done. So the idea is every time you drag into a new quadrant, you're thinning out your bacteria more and more and more until hopefully you get individual bacteria cells, which then grow into a few million, and you can look at what we call colony morphology and characteristics. So I'm going to go from here, drag across this guy, go back and forth, something like that. Flame my loop again. And keep doing the same thing here. So you're going to pull from this across here, flame it, and then pull from here across to here. And hopefully by the end of that, we should get nice colony isolates. Oh, I'm going to do a better job streaking. Gotcha. Oh, that's not going to be it. I want a few more bacteria on here. There. Okay. Good there. And one last time, we're going to drag across this guy. And so again, the idea is not to get as much as possible, it's to thin it out over time. Then you're going to incubate it, keep it in the incubator overnight at 37 degrees Celsius. And look what you got. And so I did okay, right? Definitely not any isolated colonies here, right? Oddly enough, I got an isolated colony there, in my third quadrant, and I've got some in my fourth, but I probably still drug across too many here. And so if it were me, I would probably pull from this when it comes time to grow this thing, right? We might want to plate this thing again, maybe on a different type of agar that grows certain types of bacteria. There's all kinds of them we'll learn about in class and that you'll do a virtual lab on. But I might want to say, oh, well, I know it's sort of this yellowish beige bacteria that grows in little circles, but we got to figure out what's going on, right? And so we can grow it on different media, we can stain it. We would want to pull then from one of these little isolated colony circles here when it comes time to do any testing on that. We can guarantee that's one species of bacteria. We can't necessarily guarantee that this is one species. It could be a bunch clustered together, okay? That's how you do those. Don't forget to make sure you open that Flash player, and then I walked you through how to do the rest of that. Okay, good luck, guys.